Good morning, bioinformatics students. This is Dr. Steve Shield. I'm still at it. Some of you have been having trouble with this opening set of slides, and it's got some good information, and I need to let you know basically what's in there and what needs to be done. So let's give this another shot. Bioinformatics, what is it? Well, in a certain sense, it's data mining the language of life. And in all life form contains DNA. Bioinformatics, that's a blend of computer science, biology, and mathematics all put together. Of course, we're looking at bioinformatics from the point of view of computer science or somebody in information systems. So to us, the computer science definition, what is bioinformatics? It's the application of computer technology to the management and analysis of biological data. Sometimes some people talk about computational biology, and maybe to a biologist they might think this is bioinformatics, but it's really not. Computational biology is just using mathematical models and information techniques to solve, biological problems. What makes bioinformatics different is bioinformatics has machine learning, it has algorithms to handle huge amounts of data, it has an AI component to it. So, different animal. Another thing you run into once in a while is called DNA computing. And that's where we try to build models based upon how DNA works for practical use. Here, if you look at this graph, you can see that somebody's using DNA molecules to be able to record a plan for flight between different cities, Chicago, Boston, and so forth. What is biology? Well, you all know that, the study of living organisms. And then, of course, the question should be, why should computer scientists be interested in biology? This is what we're going to kind of hopefully get out of this series of slides. What can I say? Last century, the 20th century, the big deal was splitting the atom and how that changed how mankind lived and made war and so forth. Well, this next century, our current century, it's bioinformatics. Bioinformatics will have a dramatic impact on the way we live and things that happen to us. So, it's something that we need to know about, and it's something that's important. Now, if you look here, here's some DNA, and you can see it, starting up there with that C and ending with the A, and it's a lot of different information embedded in there. This is a digital thing. Instead of having 1, 2 binary, basically it has base 4. Every position can have either an A, a T, a C, or G. The challenge is to write a program that could identify where the start of a gene would be. So if the green area represents a gene, and ATG, that's right up here, represents a start, which is the true start? So the real challenge here is to, find out where the gene is. And can, we as computer scientists help the biologists and the geneticists find that information out. Here you can see possible starts. Down here you can see possible stop words. And where's the true gene? Is it really located here and we have 2 separate genes? Or do we have a gene here, some junk DNA in between, and some materials down here? So that's the challenge. So basically what are we saying? Does the language of life have a structure? Does it have a grammar? And this is, one thing that we want to look at. And of course, German has a grammar, Java has a grammar, English has a grammar, Spanish has a grammar, and, scientists are trying to discover the grammar, the language behind the language of life, behind life itself. And it's a challenge, but we're making progress. Now, let me see. This slide, I think, brings some DNA. Here you have some DNA coming in from the left. You probably recognize what that is. Let me show you what it is. Aha! It looks to me like it might be Dr. Shield, a robust young man, taught at Coastal many years. Just obviously with that smile, he's a nice guy. Okay. Now here's a friend of Dr. Shields. The DNA is coming here at the bottom. Let me see, where's my friend? Ah, there he is. And let's do an experiment. Let's say take some DNA from this guy and take some DNA from this guy and merge it together. So let's see what we can do here. Okay, I'm taking some DNA here and some DNA there, and let's see, we'll merge those two DNAs. Let me see, where did the DNA come from? Well, you can see that the genes and stops, and we're borrowing DNA from both organisms, and we're going to blend it in together in a experiment and in some clandestine lab, some hidden off someplace. And see, what do we produce? Aha! We're going to produce something like this. Well, it seems to be like it's part human and part orangutan. Can we do that yet? No. But we can make a mouse that has human brain cells in it to test for brain cancer. That's a pretty smart mouse. So, bioinformatics has the potential for some good and perhaps some bad. Next slide. Oh, yeah, this is the one I put up on the Facebook. Couldn't resist it. National Geographic, The Missing Link, Dr. Shield embedded in that picture. A revolutionary discovery in Horry County, South Carolina. Okay, so here we'll go on to the next slide. Okay, eukaryotes. Eukaryotes are one family of life. These are organisms such as animals, plants, fungi. Genetic material is organized into a membrane, bound, or nuclei. Genes represent a small fraction of the DNA. That makes us belong to the family of eukaryotes. And you can see that eukaryotes, have a wide range from mammals to plants to fungi. Okay, another form of life is the prokaryotes. Now, the prokaryotes are your bacteria and your viruses. Now, in the eukaryotes, the genes are just a small part of the total genome. A genome is the entire DNA for an organism. Prokaryotes are different. If you look at this, the genes occupy about 80% of the genome. And so, they don't have a distinct membrane. And, that's how viruses work. When you get a virus, it's going to use your genetic material to replicate. A third form of life that we didn't know about for a while was archaea. These are organisms that live in extreme conditions. Places that were so salty that normal life couldn't live, places where they can't get air or extreme heat. They were first discovered and identified down in the ocean when we were looking at thermal vents at the bottom of the sea from an underwater volcano, and we saw this life form living right, with the scalding hot water was coming out, the material, and this is the archaea. So there are three families: eukaryotes, prokaryotes, and archaea. Now, as we got to understand DNA, there was a race. This is a picture of Francis Crick, Nobel Prize winner, him and his buddy. They were the first ones to figure out the double helix chain. There was a big race around the world to do this, and the United States, Dr. Linus Pauling was almost there. And what they were doing was they were putting together information from different sources. So what these guys, although they didn't completely do the research originally, they did some of it, but they saw suddenly insight into everybody's work and was able to put it together and finally come up. So it was a big step forward for mankind. A protein. A protein is a large molecule made up of a chain of amino acids. So this is part of the vocabulary. So you can see this is a double helix. There's genes. Genes contain instructions for making proteins. That's one definition of a gene, is a protein engine. You can see that the proteins are shaped in different sizes. And if you watch that information in a cell video that I put on Facebook, It shows you how the inner workings of a cell work. Amino acids. Amino acids are, the building blocks of the proteins. But if you look at each amino acid, there's 20 of them that have been identified. Each amino acid is a word in the vocabulary of the language of life. So if you look at the ALA in position 1, it's made up of 3 nucleotides, G, C, and A. And remember, at the very base level, the nucleotides either can be a G, a C, an A, or a T. DNA, that contains the information on you. I mean, it's your nuclear acid molecule form. It's the double helix. It's the blueprint from what makes you, you. Now, I'll end this slideshow right now. This is part 1. We'll continue with part 2 in the next slideshow. Greetings, Bioinformatics students. This is Dr. Steve Scheel. Trust you had a great Labor Day weekend. Mine was too short and too busy as usual. Let's get back to looking at some of the vocabulary associated with bioinformatics. Here again we have the double helix. Remember, that was discovered, the structure of that was discovered by Dr. Francis Crick and his colleague back in the 1950s. And this is the major component of all the language of life. If you look at your length of your DNA, your genome, it's over 3 billion nucleotides long. Your body has 100 million cells. What's amazing is that each individual cell contains your complete genome. If you unravel the DNA in just one of your cells, it'd be over 6 feet in length. If this weekend you unravel your roommate's DNA, it would stretch back from the sun and to the earth 600 times. We were amazed to discover that humans only had 30,000 genes. We thought we were so important that we'd have over 100,000 genes. But our genes have, our special purpose genes that have multiple uses, multiple functions. Good news, or maybe it's bad news. Your DNA is 99.9 some percent identical to mine. That little bit of difference makes you, you, and me, me. So the basic building block is the nucleotide. The A, the T, the C, the G. They make up, the nucleotides make up the words. The words make up amino acids. The amino acids make up the protein sequences. So the nucleotide is the foundation of the RNA and the DNA. Another important word is a gene. Scientists have many different definitions for a gene. I think I've seen over 10 definitions. One definition we can use in this class is a gene as a hereditary characteristic, a unit of heredity. Another definition of a gene that we're going to be using quite a bit is a gene as a protein engine. It manufactures proteins that the body needs. If you're in Walmart today or tomorrow and somebody asks you, what's a codon? Here's your answer. A codon is a word in the language of life. It's 3 letters long, 3 nucleotides long. It can be made up of A's, T's, C's, and G's. Now here you see, if you look closely, right here, GCU. Where did the U come from? Well, when we refer to RNA, we substitute uracil, U, for the T. But basically they have the same structure, and for this course we'll consider them virtually identical. The genetic code. This is an important breakthrough as well. This table. This relates the DNA to the amino acids. And remember, a codon is a word made up of 3 nucleotides. Those codons code for a particular amino acid. There may be 64 possibilities, 4 times 4 times 4, for 3 letters long, but there are only 20 amino acids found in life. So your genome, that's all the DNA found in the organism. And remember when we watched that video on the Facebook site for the complexity of the language of life, this is what we find here, something highly complex. One term that we come across once in a while is an open reading frame. That's an area of interest where we suspect that there is a gene in there. And over here you can see the word ATG. That's what we would call a start codon. But there's another ATG right there. So that's what's frustrating when we're dealing with DNA. It's highly complex. There's no spacing, commas, question marks, exclamation marks. Once in a while we have start words, and once in a while we have an ending word. Here's an end word, TAG. But we can't assume that everything between here and here is part of the gene. Here's an example. Here's a gene represented in green, and it comes in 1, 2, 3, 4, 5 different parts. But, if this was a start, ATG, and this was a start, and TAA is a stop here and here, which is the true start and which is the true stop? This is the complexity that the scientists are faced with, and this is why there's a great need for computer scientists and people in information systems to be able to write, design software systems, implement systems that allow the scientists to decode this very complex work. A frame shift. A frame shift error can occur when a letter is dropped out in a communication process or in a cellular process. Here we see the correct translation, ATG, AGA, TAG. But if this G is deleted, that A over here shifts over and we get ATA, GAT, AG. This can be a very serious error because it throws off all the words from this point forward. So a frame shift is caused by a deletion or an insertion would also cause a frame shift. Give you an idea of how this works. Suppose I had the sentence, how the man won the new red BMW car. If we drop the W and then try to pronounce the message that we're going to get, remember the T shifted over, we get hot him all ont hen ir ebd mimber r. You can see the problem there. Here's one of the biggest challenges. The information is rapidly doubling over in a year's period. In other words, it's growing exponentially. And we need more powerful computers, more powerful databases, more powerful programs to be able to analyze this data so that the scientists can make sense of it. What's the biggest challenge facing bioinformatics? To make sense of vast amounts of genomic data. That's it. Here you have an old cartoon that I've seen around once in a while, but I thought it was appropriate here. And that's why we need a computer. Well, that's how it is with bioinformatics, the complexity. Biological databases. They contain many different types of information. They can maintain annotation on the DNA and the protein sequences. What are they? They can also talk about their structure. And they also can talk about their expression, what they do, what's their function. So the big challenge is you got different size databases, you got the nature of the different databases, different distributions around the world. So it's a big deal. Then you have some of the specialized databases, the three-dimensional models and the structures, so forth. So it's really complicated, and obviously it needs computers to be able to make sense of this. Why is organization important? Well, we've already said that. Because of the wealth of genomic data. The data is all over the place. The data is being increased every day. The data should be provided in a clear, consistent manner with some visualization tools to aid biological interpretation. That's the goal anyway. Major analysis tools, well, there's similarity searching and protein function analysis. In other words, what's similar, if I have an unknown sequence, what is similar to it? And, I have, if my unknown sequence, what might be the function of my unknown sequence? Similarity searching tools, we were looking for things that are similar to our unknown sequence. Because if we find those things, their structure might be the same. They might have the same function. So we take an unknown sequence and say, is there anything out there that matches this or comes close to matching this? Again, protein function analysis. What is the function of this unknown sequence I have? Is there something out there that matches closely to this that might perhaps have the same function? That can save the scientists a valuable amounts of time and research if we have good programs. A motif. Well, here, a motif. This vocabulary word it's a pattern of nucleotides that has biological significance. Doesn't have to be long, but if you look over here, you could say, well, all those red nucleotides in these organisms are the same, right through here, here, here. You can see some of the patterns. So motif is a biological area of interest. It has some kind of biological significance. Let's stop this video for now, and we'll continue with the third part later on. Good morning, students. This is Dr. Steve Scheel. This is our part 3 of our Introduction to Bioinformatics. Here we're learning some of the vocabulary and some of the concepts behind the subject. Don't worry if you don't have a good handle on all the topics. Some of these things can be complicated, but a lot of this we're going to be learning as we go throughout the semester, so it's not something that should be frightening at this point. One of the words we were talking about last time was motif, and a motif is just basically something of biological significance. I mean, it can either be a nucleotide sequence or amino acid sequence. In this diagram, you see that maybe we had one sequence that was unknown to us. We discovered it, and then we compared it with 3 or 4 other organisms, and the software identifies the areas where they have something in common. And you can see the motifs are the areas that are colored, that are in columns that are all the same colors. So those are the motifs, something of biological significance. When we think of the uses of bioinformatics, there are many. One is the more drug targets. In other words, medicines that are specialized for a particular ailment or disease and that are targeted just for that rather than a broad spectrum. Personalized medicines, medicines that use your DNA and the medicine to target a particular disease. In other words, instead of seeing all these commercials with the side effects, here we're seeing that we can actually target your ailment without the side effects. Preventative medicines. We're just, we have some broad medicines that prevent the flu and so forth, but here, we're looking forward to the future where we can, really target a specific illness and keep it at bay. Gene therapy. Again, can we modify our genes if we're born with a genetic defect, a genetic ailment, a genetic disease? Can we come up with a way of modifying our DNA so that we no longer have that particular problem? Another use of bioinformatics is microbial genome applications. Well, waste cleanup. In other words, what if we had a artificial life form built on DNA that ate oil and gave off oxygen? That would be, what the world would go pay dearly for that kind of thing. Climate change. Can we modify the climates via bioinformatics, plant life, and so forth, and improve the quality of a particular part of the earth? Can we turn a desert into a garden? Alternate energy sources. Again, we're living in a day where Everybody's talking about energy and energy production. Can we come up with an alternate source based on studies from plant life or animal life in bioinformatics? Biotechnology, antibiotic resistance, forensic analysis of microbes, the reality of bioweapon creation. I mean, that, you know, we have governments working on bioweapons all the time, and this is, for good and bad. I mean, could we develop a weapon that only picked out a particular type of person and wiped our enemy out based on a biological profile that they carried? I mean, it's we really don't want to go there, but it is real. It is something that we have to deal with. Evolutionary studies, the studies of how animals and organisms are related via evolution or the design. Agriculture, can we improve crops? Now, can we make them insect and drought resistant? Can we improve nutritional quality? Can we grow crops in a poorer soils? I mean, in other words, have a crop that just takes to the desert and grows. So, these are areas of genetic research that bioinformatics can help the scientists with. There's a long, long list of companies that are interested in bioinformatics, from Eli Lilly to Procter & Gamble and Glasgow and Monsanto, Johnson & Johnson. Procter & Gamble in Cincinnati hires a ton of computer scientists that are familiar with the concepts of bioinformatics, and basically, they employ them in their genetic research. In vitro, a Latin term meaning in the glass. Initial studies for drug effectiveness are first conducted in vitro, in the test tube, in wet labs, with specimens. But more and more bioinformatics has taken us to in silico, an expression used to mean performed on a computer or via a computer simulation. Chiang Mai, Thailand. I had the opportunity to present a lot of these concepts at a conference in Chiang Mai, Thailand at Chiang Mai University. And here you see the three of us. I'm the one on the left, the handsome guy. In between is a very intelligent elephant. He actually picked a dollar bill out of my pocket. And next to him is Dr. Jean-Louis Lassalle. He is a world-class computer scientist that we had at Coastal for about 6 years before he retired. And he is the one that really got me interested in bioinformatics as a sub-area of computer science. He is in big demand all over the world. He speaks in Europe and Japan and Australia and China and Thailand and wherever. So we're back to this idea. Does the language of life have a structure and a grammar? And the answer is yes. But there's a lot out there we don't understand. And, this is the big thing. Can we help, decipher the language of life? Languages come in many different forms. Don't let this slide get to you. But, here you have an alphabet, A, C, G, and T. And you have examples of 2 languages here. And, one language, I ask you to, well, let me go back up to that. One language, I ask you to look at the sentence ACTGAC and parse it. Tell me what words are here. Well, somebody might say AC, and another person might say TGAC, the first word and the second word. But somebody else might come along and say, well, no, it's the third word, ACTG, and the first word. So languages can be ambiguous. And so it is, the complications involved in amino acids and nucleotides in the language of life make it a real challenge to decipher the language of life. Brings us to a philosophical concept. What is the first cause? What created the Big Bang? Is it mind that created mass, or is it mass that created a mind? Again, the philosophers, the scientists can debate this on and on. DNA enigma. The mystery of the origin of the information needed to build the first living organism. What is the origin of biological information? You know, this perplexes many scientists, is how the process got started. Bill Gates, multi-billionaire. Coastal Carolina University has produced 2 or possibly 3 students now in the computer science department or information systems that have gone on to become millionaires. One of our students lives on the same lake as Bill Gates does. Of course, he's just a millionaire, and I'm sure billionaires don't speak to billionaires or vice versa. But here's what Bill had to say. DNA is like a computer program, but far, far more advanced than any software ever created. DNA contains the digital instructions for proteins to make proteins. It follows that building a living cell in the first place requires assembly instructions stored in the DNA. And here's the conundrum, the puzzlement. You and I are a digital device. You know, every position of those 3 billion letters can contain an A, a C, a D, or a G. That makes us digital. But I claim that we are wonderfully and fearfully designed. Well, anyway, that ends our introductory slide series. More to continue. Have a great day, and if you have any questions, make sure you contact either Danny or myself. Bye.